Hi guys, in this video lecture we are going to discuss about cell cycle and regulation of cell cycle. A cell to grow or proliferate, it needs to undergo cell cycle. So cell cycle consists of two phases, or cell cycle completes in two phases, that is interphase and mitotic phase. So first is interphase. The next one is mitotic phase. So in interphase, now interphase is again subdivided into three phases, that is G1, S and G2 phase. G is called as gap, that is gap 1 phase, and S is called as synthesis, so S phase, and G2 is gap 2 phase. So here G1, S and G2 phase. Now the mitotic phase is further divided into karyokinesis and cytokinesis. So this is mitotic phase, this is interphase. So let's discuss about interphase. So first is G1 phase. Here the protein synthesis occur, ATP synthesis occur. Here the cell prepare itself to go to the S phase, and all the RNA synthesis occur in G1 phase. And next the cell go to S phase. S phase is the most important phase of cell cycle because here RNA replication occur. And histone proteins are formed. Now the cell pass from S phase to G2 phase. Now G2 phase also same like G1 phase, cell prepare itself to go to the M phase, that is mitotic phase. So here also protein synthesis occur, ATP synthesis occur, and tubulin proteins are formed, because tubulin proteins are required for spindle fiber formation, and here in G2 phase tubulin proteins are formed. Now after completion of interphase, because from here to here it's interphase, now the cell passes to the second phase of cell cycle, that is M phase. So M phase is also called as mitotic phase, and it comprises two phases again, that is karyokinesis, cytokinesis. What is karyon? Karyokinesis means nuclear, karyon means nuclear division, and cytokinesis is means cytoplasm division. So karyokinesis and cytokinesis. So in karyokinesis four phases are there: prophase, metaphase, anaphase, telophase, because our mitotic cell division occur here. That is, mitotic cell division is equational division, and that is different from meiosis division. Meiosis is reductional division. So mitotic cell division occur, and the four stages are prophase, metaphase, anaphase, telophase. Now after this telophase, then when the nuclear get divide, now the cell goes to cytokinesis phase of mitotic or M phase. Here our cell divides into two daughter cell, which contains the same haploid cell or diploid cell of the parents, diploid cell 2n of the parents. Now here the two daughter cells are formed. Now the cell has formed. Now there is another phase in the cell cycle called G0 phase, which is here, and this is called as inactive phase, because suppose we don't want a cell to grow, or we don't want a cell to proliferate. Here everything will occur, like cell will grow, cell will get all its activities, but it will not passes to G1 phase, or cell will not divide. And in case if we need, then from here G0 phase the cell will go to G1 phase, and all the cell cycle continues, that is interphase and M phase continues. So it is called as inactive phase of cell cycle. And so next we will discuss about regulation of cell cycle. So So when the cell goes or travel through the cell cycle, there are certain checkpoints. As you travel through the road, there are certain toll gates or checkpoints are there. In the same manner, in cell cycle also, certain checkpoints are there which will check whether the cell has ability or cell have acquired all the proteins to pass from one phase to other phase. And that checkpoints are, we have three checkpoints. So first is G1 checkpoint, G1 checkpoint, and second is G2 checkpoint, and third is M checkpoint. So here this is G1 checkpoint, and second is here the G2 checkpoint, that is at the end of G1 cycle. G1 checkpoint is there at the end of G2 checkpoint, at the end of G2 cycle or G2 phase, G2 checkpoint is there, and at the end of metaphase, this is prophase, metaphase, anaphase, telophase. At the end of metaphase, this M checkpoint is there. M checkpoint is also known as spindle checkpoint, and we are going to discuss all this checkpoint in a minute. So this G1 checkpoint, it will check whether the cell or in this G1 phase all the synthesis occur or not for the DNA replication, because for DNA replication we need ATP, we need mRNA, tRNA, all the RNAs, and we need all the proteins. So G1 checkpoint will check this G1 phase, and if everything is okay, then it will pass the cell from G1 to S phase, and S phase the DNA replication, and next it will pass to G2 phase, from S to G2 phase. Now G2 phase also, here there is another checkpoint that is called as G2 checkpoint. Here it will check whether the DNA is replicated properly, because in S phase we have already completed our DNA replication. So here in this phase it will check whether the DNA replication occur or not, or whether all the proteins which are required for the next M phase it is there or not. And once everything is checked, then it will pass the cell from G2 phase to M phase, and next the mitotic division occur. And now another checkpoint, the third checkpoint, as I told, that is M checkpoint or spindle checkpoint. Because why it is called spindle checkpoint? Because it will check the spindle fiber, whether the spindle fiber has attached to the kinetochore of the chromosomes. Because at the anaphase the spindle fiber contracts and both the chromosomes comes apart to the two different pole of the cell. So that's why it is called as spindle checkpoint, because it will check whether the spindle fiber has attached to the kinetochore of our chromosomes. So these are the three checkpoints. And for who regulates these checkpoints? So for regulation of this checkpoint we need a protein called cyclin and a kinase which will phosphorylate different proteins for our cell to pass from different phases. And CDK. CDK is cyclin dependent kinase, because this is the cyclin protein and this is CDK. Normally CDK is always present in the cell, always present in the cell cycle in an inactive form. But when cyclin will bind to the CDK, suppose this is the CDK and this is in inactive form now, and when this cyclin will bind, suppose this is cyclin will bind, now it will get activated, and this cyclin plus CDK complex are required for every phases of the cell cycle, so that it will pass the cell from one phase to another phase, one phase to another phase, G2 phase to M phase, and so on. So this cyclin and CDKs plays a major role in cell cycle or cell regulation of cell cycle. So here in G1 phase we have cyclin D and cyclin D plus CDK4 and CDK6 are required in the initial phase of G1 phase. Now at the late phase of G1 phase, cyclin E and CDK2 are required, because this cyclin E and CDK2 will combine and it will activate sorry, it will activate the CDK, because CDK priority is that it needs cyclin to get activated. Now CDKE plus CDK2, sorry, cyclin E plus CDK2 will combine and it will get activated and it will pass the cell from G1 phase to G2 phase, and this is called as positive regulation, because here the regulation is of two types: positive regulation, negative regulation. Now in the same way sorry, in G2 phase also we have another cyclin and CDK, that is cyclin A and CDK2. In M phase also we have here cyclin B and CDK1. So these are all the positive regulation. But now when there is any DNA damage is there, or when the cell is not prepared itself to pass from G1 to S phase sorry, when there is a DNA damage is there, or cell is not prepared itself to pass, or enough protein is not synthesis which cannot pass the cell from G1 to S phase, at that time there is negative regulation occur. So regulation is two types, positive and negative. So let's discuss the negative regulation. So when there is a DNA damage, now there is a protein called P53. It is called as guardian of genome because it will guard all the genome of the cell. That's why it is called as guardian of genome. This DNA damage, when there is a DNA damage, this P53 protein will get activated, and it, as we know, in apoptosis, intrinsic pathway is activated by P53. So it activates the intrinsic pathway of apoptosis. Now cell death. So first initially it will arrest the cell to pass from G1 to S phase, and it will prepare the cell. So first it will check and it will prepare the cell, and if not, its cell is not repair, that is in prepare phase, the DNA is not repair, then it will go for cell death or apoptosis. So another function of P53 protein is it will activate the transcription factors, and it cause production of P21, same as P53 is P21. What it will do, P21 is an CDK's inhibitors. It is an inhibitor. It is an inhibitor of CDK. As we know, CDK, that is cyclin dependent kinase, is required for passage of cell from one phase to other, and CDK inhibitor is P21. Because is there any cell damage or DNA damage, we don't want a damaged cell to replicate or damaged cell to divide. So here P53 protein, what it will do, it will activate the transcription factor and it will produce P21. This P21 protein is an inhibitor of CDK, and thus CDK is inactivated and our cell get arrested. So this is the complete scenario of cell cycle and the regulation of cell cycle. I hope this video will be helpful to you. Thank you.